I would like to present recent work on vector representation for phylogenetic trees. So it's a joint work with two colleagues in Simon Fraser University, Cedric Schaufer and Caroline Collins. So the phylogenetic trees are essential model for the evolution of genes and genome. So they play a very important role in human evolution, the study of the dynamics of viruses. They're also useful in investigation into the language evolution. So, however, the inference of these trees is very challenging. So the programs are time-consuming, prone to errors. This is a popular method. So IQ-tree, Rex, ML, the BEAST, all these tools are based on the following search strategy. So basically, you define a neighborhood relation in the tree space. Then based on such a relation, you traverse the tree space from one tree to the neighboring trees, stopping at the local optimals. So according to an objective function. So such a strategy requires a good representation for the trees. So basically, we need to have the representation that can be used for faster processing or efficient storage. So the popular one is a NOIC format. So it's widely used for storing trees, okay, but not for processing. So in this format, so each pair of OP bracket represent an internal node. So therefore, so the representation contain n-1 nested OP bracket if you have an in taxa. So this is optimal for storage purpose. But recently, a group of research from Oxford University investigation another representation. So this, each tree is represented by a vector of n-1 dimension. So this is actually based on very simple factor. So each tree can be generated from a single leaf tree by adding the leaves one by one. So say, for example, this is a 5 taxa tree. We can generate it start of one single leaf tree. Then we add another taxa to the unique edge. So the A1 actually specifies the edge. So then we add a third one. Also, there are three possible edges to add. So A2 specifies that edge. So and so on and so forth. So this is another representation they call philo to vector. So this is what I'm going to present. So in this format, so each tree, binary tree is represented by a string of 2n characters. So basically, each character appears twice. So we use the index of taxa. So this the representation is based on the tree decomposition technique using the study of phylogenetic networks. So in our recent work. So this is the idea. So basically, we have this binary phylogenetic tree. That if we have ordering of the taxa, so we can take any ordering. For this example, we take alphabetical order. And then we can decompose the tree into a disjoint process. This is quite easy. So basically, we consider the smallest taxa first. They would move up from that leaf up. We will reach the root. So this is our first pass. Then after that, we consider second one. So we move up from the second one, eventually we will reach the first pass. When we reach that there, we just stop. So this is our second pass, and so on and so forth. So we decompose the tree into these disjoint passes. So each pass starts with an internal node. They terminate in the leaf. So therefore, we can label the starting internal node using the taxa labels. Okay, so therefore we have these labels for internal node. We have a path, we have a labels, then we obtain streets if we work down the path. So therefore, for each taxa, we have a string. Conversely, so we can combine all the passes to obtain the original tree, because the label information gave us how to glue the passes together. Okay, so therefore then we just put all these strings together, so we form the vector. Okay, so the important information is these strings. So the black one characters are important information. So the red character actually encoding the ordering we use. Okay, so if we use alphabetical order, we don't need that one. So this is the characterization of the vector representation. So basically, each is a string over 1 to n. So each character occurs twice in the string. For each character t, the first copy of the t appears before the second copy of t minus 1. The second copy of t appears after the second copy of t minus 1. So there are two examples here which represent the two trees over 6 taxa. But we can also generalize this to the non-binary trees. For non-binary tree, each internal node is represented by a subset of the taxa. It's still unique. Yeah, each character still appear twice. Okay. They can also generate to a class of networks. So you see, this representation is quite efficient. So this is how to decode the tree structure from the representations. Basically, you see, for each internal node, there are two outgoing edges. So from the C on the top, there are CB, there are CE. So you see the CB is within the path. So therefore, if we read the string from left to right, we can get this edge directly. So another, the other edges are between the node in two different paths. So there's like a CE. So the C is the green path, E is the purple path, in the purple path. So for this, we can also find out this path by reading the string from left to right. It's just a little bit difficult. Okay, so this is a decoding. So also a linear time decoding algorithm. As I mentioned earlier, so we use this for inference the phylogenetic networks. But here I mentioned another application. So basically, we can use this to define a new metric for tree comparison. So first of all, we have this hop operation. So basically, we just move a first copy of a character t to any position before the second copy of t minus 1. We cannot move this beyond the second copy of t minus 1. So actually, this is a SPR operation. See, for example, if we hop on, we do the hop operation on the taxa 3, then we move the 3 from the second position to the fourth position, just before the second copy of 2. So actually, this is equivalent to this. We caught the middle tree they move this middle tree to another position. So in other words, this vector representation allow us to select a set of SPR operation to do the tree transformation. So this is, you see, it's quite efficient. These 15 trees, we need at most 2 operations can reach from the middle one to every other one. So then we can define the hop distance. So it's a minimal number of hop transfers used to convert one tree to another. But we have this surprising result. The hop distance can be computed in n log n time. They also, the distance between 2 any trees is at most n hop operations. It's quite efficient. So how to do this is very simple. So we have a tree representation. We just do the sequence alignment. Okay. But these are special strings so that we can do alignment in n log n time rather than in square time. So therefore, we argue that this operation is very efficient. The distance is also easy to compute. Another application is this. Basically, we can calculate the transformation between the 2 trees, but this calculation also reveals the common patterns, common structures in the 2 trees. See, for example, this is 2 large various trees. If we calculate the Robinson foot distance is 120, so it doesn't mean too much. But if we calculate the hop distance is 24, then also we can recover the large subtree, so which contains 34 various samples. So it's showing the right. Okay. So I think I think that's all. So basically, we introduce a new tree representation. It has some nice properties. I think so. We think so. Using this, as I mentioned earlier, so we define a hop distance. So this is this distance can be computed in log n time. So this is very rare. Okay, normally when you define the distance based on the tree transformation, then the distance is in p hop. Okay, so also I think this is useful to compare the large trees. So this is work done in May. So the manufacturer is available. So sorry, the manuscript is available on arXiv. The program for decoding, encoding, the computing the hop distance are also available on the GitHub. Okay. Okay, so here is some research problem. The first of all, is that this is a weakness of this distance. So basically, the hop distance depends on the taxon ordering used in the definition. So we use the ordering to flat the tree into a string. It's something like we do the dimensionality reduction in the big data analysis. We lose something, but we provide a faster approximation approach for solving some problems. Okay, so therefore, in theory, so we can define the maximum hop distance by consider all possible ordering. Now, if we do it in this way, that we really don't know how to calculate this maximum hop distance. Also, we don't really know the upper bound on the hop. So this distance. So this is just a theoretical studies. So this is the last question is the most important one. So we just how can this new representation be used to develop faster programs for tree inference and tree clustering? So this it may take a long time to answer this question. I think this is the most important one for, at least for us. So I think that's it. Thanks.
Thank you for your talk. I like the idea of being able to quantify and compare the tree similar structures.
Yeah.
Could I use your method for cancer evolution, cancer subpopulations?
Cancer, basically, we already have the labels on the internal node. It's a little bit difficult, but we also propose some decent measures for the cancer trees 2 years ago. Yeah, but this one I find it could be used for compare the RNA structure that I just talked in the first talk, because we can use this for non-binary trees.
Thank you for your talk. It was very interesting. I was wondering whether your representation could be used to facilitate the analysis of gene trees using machine learning.
Yeah, this is a motivation for this work. So, yeah, thanks. This is nice.